Good morning, everybody. Am I muted? No, I'm not muted. Cat. Okay. I'm gonna sort myself out. Got this. And just making sure I got all the PowerPoints open that I should do. Hey, that's the other one. Okay, let's again. Yeah. Well, sometimes, you just have a cup of coffee because you want a cup of coffee. Most times it's because I need a cup of coffee. Okay. All right. So let's have a quick look at the schedule. Make sure I keep my mind on things. All right. So essentially, what we've got to do this week is finish up gene regulation. And then we've got a couple of kind of bigger picture things to talk about. Biotechnology, genomics, essentially kind of taking all of the things that we've been learning about and putting them into context, which is always fun. So, and what we're going to do today is finish off prokaryotic gene expression, which we did a bunch of. Oh, geez, what day is it today? Tuesday, last Thursday. I'm kind of a bit spaced out, to be honest. And we can recap a little bit of what we covered on Thursday too, because it doesn't hurt to do so. And then most likely in the second session, we'll start on eukaryotes, which are pretty crazy, right? As a general theme. I don't know whether hopefully you're not going to go into micro after this and tell Dr. Russian that prokaryotes are like super simple and eukaryotes are where it's all at, because otherwise I'll get in trouble. Because prokaryotes are pretty cool. They do lots of really kind of fun, neat, and somewhat alien things, that's for sure. Anyway, so let's bring up the PowerPoint. And this is a good time to ask questions too, right? So a good practice would be and you're going to have these in fall most likely, because we're only doing labs in the fall, I think, at least at present anyway, is to go through what we've covered the previous lecture and then basically make a list of questions or I don't know, and bring those to the start of next lecture. Every professor does things differently, so there may be a greater or lesser opportunity to do that, but it's generally a good idea, right? Because office hours obviously are very hard to have, right? And these kind of count as office hours in a sense as well. Oi, leave me alone. So make the most of that too, right? So essentially, anybody got any questions about any of this craziness, right? If I were to ask you, let's do it actually, is, hang on, let's put it up here. I don't know if that's any better. Is the lac operon inducible or repressible? It's opposite. It's inducible. Good guess though. So inducible means that it's normally off, right? So there's a lot of, don't know if anyone's read or watched 1984 by George Orwell, but there's a lot of doublethink in biology, right? So repressible doesn't mean it's normally, well, repressible means that it's normally on, right? And it needs to be repressed to be turned off. The trip operon, which we'll be covering in a little bit, is repressible. What do you need for the lac operon to be expressed at all? What has to be present? Clues in the name, lac. What does lac, what's that short for? It's a metabolite. Find it in cow's milk. Oh, there you go, lactose. So is there any point expressing the lac operon in the absence of lactose? There isn't, right? Because, you know, what's the point spending all that energy, metabolites and resources producing proteins to import and metabolize something that isn't present, right? There's no need whatsoever, right? So the lac operon is inducible, right? That means that it is always off until it's told to turn on, right? That's a really important concept to get, right? Typically, that's the case for most eukaryotic genes. Most eukaryotic genes are off and have to be turned on, right? So the lac operon is inducible and the inducer is lactose. Now, what keeps the lac operon off normally? Is it just normally off, or is there something that actively stops it from being expressed? If so, what is it? These are all great exam questions, by the way. That's it. Awesome. Right. LAC-I, which is a protein that binds. Oh, there you go. What does it bind to? What part of the lac operon? It's actually on the page right in front of you that I'm showing you. Actually, no. Good answer, but not quite correct. What binds to the promoter? What's the promoter for? Promoter is where the RNA polymerase binds, right? The operator is where the regulatory protein, pardon me, in this case, LAC-I, binds, right? So always remember, get out of it. Regulatory to the operator. Should I bring that out a little bit? Yeah, I did spell that right. Right. And typically, but not always, the operator is in front of the promoter, or downstream is probably a better term. Right, and so you can see how anything binding to the operator stops transcription, right? The RNA polymerase is not physically able to get through. Sometimes it can't even bind, right? Because regulated like LAC-I protein may actually physically block it from binding to the promoter, right? Even if it can bind, it can't get through because there's a protein in the way, right? It's blocked. It's like some large kind of rock fall has come down and blocked the road. Okay, so when is LAC-I normally bound to the operator? Under what conditions? This is the same question I've asked before, but just from a different perspective. Absence of, I'm not sure what lack of lactose means, but absence of lactose, right? So when lactose is not present, lac-I is bound to the operator. That's why it's, yeah, I figured as much. That's why the lac operon is an inducible operon, right? Essentially, the lac-I has to be induced, right, to leave, right? And that's what lactose does, right? So when lactose binds to lac-I, it causes a change in shape, and essentially the lac-I is no longer able to bind to the operator. So would you think that would be the large scale change in expression or the small fine tuning change of expression that is common in prokaryotes? Which one do you think that would be responsible for? No, that's the big one, right? So you can't fine tune expression when no expression can occur at all, right? So removing or binding a regulatory protein to the operator is big, right? It essentially goes from like all out potentially to zero, right? It's a very binary switch. Either works or it doesn't, right? It's like you can either get through on that road or you can't. Or in San Antonio, it's like, it's only a little bit flooded. We'll try anyway. And then your car dies. But that's a bit of a digression, right? It's either blocked or it isn't. So it's large scale tuning. I actually had a student in my GenBio 1 class once. She's like, I thought it was shallower than it was. And then a brand new car basically got totaled as a result, which was rather a shame. Anyway, okay, so what is responsible for the fine tuning of the lac operon, right? So this isn't about is lactose present or not, because the lac operon is being expressed. So lactose has to be present, otherwise it's going to be off. So what's responsible for kind of tweaking the level of expression to match what is needed, right? So you can think about this in lots of different ways. And this is something that I'm trying to get you into the habit of. There's more than one way to ask and typically answer a question, right? So what do we care about when it comes to fine tuning the lac operon expression? What metabolite matters now? It's not lactose. Awesome. That's fabulous. Why does glucose matter in terms of fine tuning the lac operon? What is glucose the result of? Well, it's fuel, right? Yeah. So we need it, or bacteria. Everybody needs it. Everything, every organism. But another way of asking this question is, what does lactose get turned into by beta-galactosidase, one of the lac genes, lac-Z, I think, or lac-Z if you're American. What does lactose get turned into? Yeah, actually, sorry, slap, slap, slap. Shouldn't have shook my head. That's one thing. There's another one. There's a second one. Nope. Lactose is galactose and another sugar beginning with G. Nope. Allolactose is just a fancy way of saying lactose. I don't know why they call it that. What's yeah. So you see what I mean? Like you already gave me the answer to this question. I'm just asking the question in a different way. It's the same answer, right? Yeah. Well, that's not sneaky. That's just teaching you to think about the same problem from a different perspective. No, it's not. Anyway, and that's important, right? Because you're correct, Angela. Glucose is fuel, right? You need it. It's constantly being consumed. But on the other side of the balance, the output of the lac operon in terms of protein is turning lactose into glucose, right? So you have basically like a balance or two scales, right? You have the consumption of glucose, right? So glucose concentration is constantly dropping, and you have the production of glucose from lactose by beta-galactosidase. I don't actually know what galactose does. I'll have to check it out, right? But anyway, you have this constant input in terms of the output of the lac operon, metabolically at least, and then you have a constant decrease potentially depending on whatever metabolic needs the bacterium has, right? So if you're, perhaps, it's nighttime, bacteria wants to go to sleep or whatever, doesn't need to use a lot of energy, right? Demand decreases, right? So glucose levels start increasing. Do you still want to keep going gangbusters on the lac operon producing beta-galactosidase? If glucose levels are going up, do you still need it? No, right? But you don't want to turn it off completely because lactose is still present and it's a good food source, right? So now you're going to start dialing things back a little bit. You're not going to shut them off, right? Which is what would happen if lactose was no longer present, right? Lac-I would rebind to the operator and shut off the whole operon, right? But you do want to dial things back, right? Because, it's like, I don't know, San Antonio and toilet paper, right? When this whole business hit, you couldn't get, like, we were bartering toilet paper for like dried beans and stuff. It was ridiculous. It was like a caveman economy, right? So if you're in a supermarket like Walmart down the road, you'd be filling those shelves as quick as you could because it was disappearing as quickly as it could. Now, hopefully, things have calmed down a little bit and people aren't stockpiling toilet paper for whatever reason. I've never really totally understood it personally because this disease doesn't make you defecate more, but who knows? Anyway, so you want to slow down the amount of toilet paper you're getting into the store, right? Because otherwise you'd fill up the whole storage area at the back with toilet paper. And it's exactly the same principle, right? You want to match the output of the lac operon in terms of transcription and translation and all that jazz with the demand for glucose, right? So you want to, when demand is high, right, you want to go crazy, produce lots and lots and lots and lots of lac-Z and all the other ones, whatever they're called, lac alpha and stuff. Lac-Y, I think, is another one. But when demand drops, glucose levels start rising in the cell, then it's a waste of energy to keep producing more glucose, right, by turning the lac operon on. Okay, so here's a trick question. Is glucose sensed directly? No. What proxy is used to infer the levels of glucose in the cell? And this is the same for eukaryotes too. It's something that not many people do in the summer in Texas. It's just too hot. Yeah, exactly. Cyclic AMP, right? Adenosine monophosphate. It's a very funky chemical, which isn't actually used as an energy source. It's not like ATP or anything like that. It's a cellular sensor for energy levels. So as glucose levels go up, what happens to cyclic AMP levels? Right. So essentially, we've got a teeter-totter, right? Glucose goes up, coffee or cyclic AMP goes down. Glucose drops, cyclic AMP goes up. How is that level? Coffee, Colombian actually, I think today. How is that level of cyclic AMP detected by the machinery that controls the lac operon, the fine tuning? What does that stand for, Ali? I'm just giving you time. There you go. And take time to write out, right? Right, catabolite activator protein. So what glucose levels are low, cyclic AMP levels are high. How does that affect the lac operon in conjunction with the with CAP? What's the consequence of high levels of cyclic AMP bound to CAP? No, actually the opposite. Sorry, love. It promotes the binding of RNA polymerase, right? So it's not something that prevents or allows RNA polymerase to bind. RNA polymerase will bind to the lac promoter in the absence of CAP or cyclic AMP CAP. But when cyclic AMP CAP are present, then RNA polymerase binds really, really well, and so will activate, or hang on, where's the, I'm trying to find the good one. Here we go. It will allow transcription to occur more frequently because the RNA polymerase is going to be binding more frequently and more reliably, right? So this is why it's a, it's kind of like a dial, right? So it boosts the efficiency of RNA polymerase. It doesn't make it work or make it not work, right? In a sense, that's what LAC-I does, right? So if LAC-I is there, RNA polymerase is not, right? And you can actually see how the RNA polymerase, this is a nice picture actually. If you kind of, let's see if I can zoom in here, right? You see how the RNA polymerase actually overlaps on the LAC-O operator. So now you can see that if there's LAC-I bound to the LAC-O operator, RNA polymerase isn't going to be there, right? Cyclic AMP CAP doesn't prevent or make RNA polymerase bind. It just makes it do so better, right? So when glucose levels are low, cyclic AMP is high. This is actually one of the trickier bits to get about this whole system, to be honest. Yeah. When cyclic AMP levels are high, we get lots of CAP-CAMP on the, not the promoter as such, but a bit of DNA upstream, which promotes the binding of RNA polymerase and therefore transcription, right? So it's like a boost, essentially. It's kind of like, you know, drinking a few Red Bulls before a study session, right? It's not going to make you learn stuff or not. It's just going to hopefully help you do so more effectively, right? Or for longer. Right? And so when glucose levels start going back up, cyclic AMP drops. There's not much cyclic AMP CAP or CAP-CAMP present. And so RNA polymerase comes in and sometimes binds, sometimes doesn't, right? So there will still be some expression of the lac operon, but it will be a much lower level, right? So it's not binary like LAC-I, it's not on or off. It's kind of somewhere in between those two. Does that make sense? And it's also a neat way of thinking about it. It's almost like a thermostat, right? If you think about it from a different perspective, right? You want to maintain a certain level of glucose. The reason why it comes to mind is my aircon just turned on, right? You want to make a certain level, maintain a certain level of glucose. You don't want it really high because that's not good. You don't want it really low because that's terrible. You want it somewhere in the middle, kind of like humans do with blood sugar homeostasis, right? And insulin and glucagon from the pancreas, right? So you want to maintain it within a level. As soon as it starts kind of going above that level, you want to decrease the production of glucose, right? Bacteria don't have anywhere to store it like we do. You know, what we do is we just store it in our muscle and our liver, right? Bacteria can't do that, so they reduce the production of glucose. When glucose levels drop, bacteria then just amp up the production of glucose if lactose is present to maintain that kind of constant level. Just the same way as we either store glucose goes up in humans and we store it in the liver, we turn it into glycogen, right? It gets taken up into the muscles, whatever, because high glucose is bad, right? So it comes back down. When it drops, because we're metabolically active, say you're being chased by a tiger or something, then insulin's released from the pancreas that basically promotes uptake. Hang on, no, it's the other way around. Dumbass. Goes up, insulin's produced, glucose gets taken into the muscle or the liver. When it goes down, glucagon is produced, another hormone, right? So glucose is released or turned into glucose, so you have it available for your muscles to take up and use, right? So it's the same kind of deal. It's just a different mechanism, right? Bacteria do it by production. We do it by storage, right? Essentially, but it's the same principle. It's like a homeostatic thermostat, right? And so kind of keep that in mind as well, because that's something that informs an awful lot of biological processes, right? A lot of them. Anybody have any questions about the lac operon? Because the trip operon is even trickier. Because this is going to be a fairly substantial chunk of the exam, these two things. Okay, if you don't have questions now, that's fine. But do please ask them either by email or, oh man, I have no idea who that is. Email or next class. Actually, that might be the assessor for our car. We got rear-ended at the weekend and it's probably going to get written off, which is a complete pain in the ass. Really like that car as well. Anyway, let's not talk about that because that's just depressing. So, yeah, more or less. Both my kids have mild to moderate concussion, so they're actually just pissed off because it means they can't stare at their screens or watch a lot of TV. Yeah, so for the parents, it's like, yes, my kid has concussion, but it means that they're going to be reading a book or doing something wholesome. But anyway, they're like a week of that. Actually, Andrew, if you could send that info to me, that would be really useful. I'm not sure we'll need one, but it wouldn't hurt to have that information and perhaps give them a call and get their opinion. Yeah, because I'm not in the habit of getting into wrecks, right? So this is the first one I've been in for, oh crap, actually since I've been in the US. The last accident I had was in Australia. Was it New Zealand? One of the two. Anyway, so the trip operon, and so again, one of the reasons I think I said this last time that we use the lac operon and trip operon is that they're really both well studied and they give you a good overview of the different ways in which we can control gene expression. So the lac operon is an inducible gene expression system. That means it's always off until it's turned to come on. The trip operon is a repressible system, right? So remember again, repress, how do you spell this? Right? So it's a very different setup, right? Kind of, and if you think about the context, right? The lac operon, no lactose, there's no point having it on, right? And your environment changes as a bacterium, presumably, based on whatever's around you, right? So you want to be able to make use of that environment as and when it changes in your favor, but any other time, there's no point, right? You don't want to express the lac operon if there's no lactose present. The flip side of that is that there are probably going to be a bunch of things which you always need as a bacterium or as a eukaryote as well, right? And so you always need those things turned on because as long as everything's going, well, then why would you not need them, right? And so the case here is now, okay, we always need this stuff. So it's always going to be on, but sometimes we may not need as much of it, right? So typically what we're talking about here is kind of commonly used metabolites or common processes, things like involved in translation, transcription, metabolism, making like cellular building blocks, things like that, right? And so we typically always need that stuff, right? In humans or in eukaryotes, they're called housekeeping genes, right? Because basically they're always on, kind of making sure the house functions, right? If we're thinking about it in terms of cells and the like. But sometimes you don't need as much of it, right? So that's what the trip operon is. And so the trip operon is all about the production of tryptophan, tryptophan being an amino acid, right? So here all automatically tells you, okay, if we need tryptophan, right, we're going to be making proteins because tryptophan's involved in protein synthesis and translation. So we always need it until, you know, we don't, right? Because we have enough tryptophan. Maybe we're not producing as much proteins anymore. So now we need to dial it down because it's a metabolically expensive thing to do, right? So we're starting from the opposite perspective from the lac operon. Lac operon is always off, only turn it on if you need it and as much as you need it to. Trip operon is always on, but maybe we need to turn it off when we don't need it as much, right? Hopefully that kind of makes sense. And that's where the inducible versus repressible deal comes in. And again, got any questions, just stop me and ask. Let's get back to, gotcha. Right, so essentially let's have a look. This is what the trip operon looks like. There are 5 different structural genes in the trip operon. I don't know why they're backwards. A, B, C, D, and E, right? These are all enzymes which are involved in a step of the production of tryptophan, right? So there's a whole metabolic pathway. Where is it? Where is it at? Do we have it here? Come back. Yeah, kind of like shown here, right? So the input is chemical chorismate, whatever that is, and the output is tryptophan, right? That's the thing that we want and need as a prokaryote. And so this whole operon encodes all of the enzymes needed, right? It's a little bit different from the lac operon, which also encoded a importer, right? We don't need to import chorismate, presumably, or if it is imported, it's going to be imported by something else, right? What the trip operon is all about is the metabolism of each of those steps of tryptophan production. Now we have an extra wrinkle here. This is where things get funky. We have the same as with the lac operon. We have a promoter, TRPP, right? Again, this is where RNA polymerase binds and it will, you know, transcribe the trip operon. We also have an operator called TRPO, you know, unsurprisingly. This is where the regulatory protein binds. TRPR, I believe, from memory. Yeah, there you go. It doesn't actually say what it's called. Oh yeah, there we go, TRPR. So that, again, kind of fits with what we've learned already, right? We've got a regulatory protein which will block RNA polymerase from transcribing the gene or even binding to the promoter if it is bound to the operator. Okay, so far so good, right? So far we're kind of, you know, within familiar territory. The brown thing, this bit here, no, the trip, we don't have any proteins on here, Angela. We're just looking at the different regions of the operon. So this area here this is called a leader region, right? That's what the brown thing is. Right. And essentially, this is the region that's involved in the fine tuning, right? So the fine tuning of the trip operon is not done with an extra protein, the cap protein, right? The catabolite activator protein and the lac operon. It's done using an inherent sequence in the trip operon, this leader region, and it's really quite tricky to get. It's not impossible, but you have to kind of like twist your head around it, right? So we'll get to that in a little bit. But just keep that in mind. Trip P and trip O, as before, RNA polymerase binding to the trip P. Trip R will bind to the trip O, the operator, and these are our structural genes. The big difference with the trip operon is this leader region, right? And this is essentially, we're going to be looking at RNA structure. Okay, so let's just head back here, make sure I haven't forgotten anything. Oh yeah, and so the trip operon is also a really good example of a negative feedback loop. So what we mean by that is that its product, tryptophan, is involved in negatively regulating the expression of this operon, right? So as tryptophan levels go up, expression goes down, basically. There's also a bunch of stuff. This is pretty much what we've already covered. But what we're going to look at now is this bit. We have an aporepressor, right? So LAC-I equals repressor. In other words, it binds to LAC-O in on its own, right? That's what we've learned so far with the lac operon. Trip R, that's P. R is an aporepressor. In other words, does not bind to trip O on its own. It needs its co-repressor. And as you probably guessed already, oh damn, this is going to be a pain. Tryptophan is the co-repressor. So now we have something a little bit different from before, right? We don't have just a straight up protein that can bind on its own like LAC-I does. Now we have a repressor which is unable to bind the trip O operator. Now it has to be complexed with its co-repressor, right? And that co-repressor is tryptophan. So now you can see how the repressible part works, right? Inducible is something's bound and you induce it to leave, right? And lactose is the inducer of the lac operon. With the trip operon, it's repressible, so it's always on, so that that trip R cannot bind, right? It's in the wrong shape, right, on its own, right? So in the absence of tryptophan, trip R cannot bind, so the trip operon is expressed, right? Only when tryptophan is present, right, as a result of the trip operon doing its job, right, and all the enzymes that it produces, only when tryptophan is present is the trip R going to form the right conformation, right? Because it's going to be bound together, the two things, trip R and tryptophan. When that happens, it can bind to the trip O operator and turn things off, right? So Let's draw this out. This kind of never hurts to draw stuff. Kitty cat. Let's stop sharing this. Oh, and I did bring my camera. Good stuff. And stop me if you have questions, please. Okay, so move that back a little bit. Essentially, we have our promoter. We have our operator. I'm not going to talk about leader sequence right now because it's not involved in the large scale turning on or turning off. Well, not turning on. It's not involved in the large scale turning off of the trip operon, right? And then we have our trip genes, right? Those are all of the genes that are involved in producing proteins or enzymes that metabolize the stuff, right? So essentially, let's draw like so. Going to be something like this, right? So when no tryptophan is present, which means that we need tryptophan, right? And essentially that's going to be because tryptophan's being used in translation, right? So it's being used faster than it's being produced, right? So there's no tryptophan present, then TRIP-R cannot bind to the TRIP-O, right? It's not the right shape, essentially. It won't recognize the DNA, right? And again, this is something that you'll hit hopefully in other classes. Protein shape is super important in protein function. It's actually one of the most important things, right? So same thing again. We have our trip on basically, let's get a different color. When this is the case, right, there's no tryptophan present, then your RNA pol is able to bind, right, because TRIP-R is not blocking it, and we get lots of expression. Right? That's essentially how that works. Now, when tryptophan is present, we've got TRIP-P, we've got TRIP-O, right? Let's use green. I always struggle spelling tryptophan for some reason. Now tryptophan is present. Now there's TRIP-R. TRIP-R here, and we have our tryptophan, right, binds to it, causes a change in shape, and now that basically binds to the operator, and now RNA polymerase can't bind, right? And that's the large-scale control of expression in the TRIP operon. Does anybody have any questions about that? Oh, and there we go. Essentially, oh, my handwriting's terrible. So you don't get any expression. It's totally shut off. Is there glare? Sorry, that's kind of my room light, which I have on so I'm not too dark. It's kind of tricky with video, trying to make sure that I'm bright enough but not so bright you can't see me. So yeah, that's basically the deal, right? In the absence of tryptophan, the protein's the wrong shape, won't bind to the TRP operator. In the presence of tryptophan, it changes shape, binds to the operator, and then prevents RNA polymerase from binding and transcribing the TRP operon. So we end up with no expression, right? That's the basic on, off deal of the TRP operon. I'll take a quick picture of that. I really miss being able to use a whiteboard, a larger one at least, because some of these things are a lot easier to draw out than they are to describe in words. But hopefully you get the gist of it from what we're doing. Yeah, I do too, Angela. I do too. But you know, time doesn't stand still, unfortunately. So we've got to make the best of it, and hopefully all of this is, you know, makes up for some of that. Unfortunately, coffee is done. I need to have I don't know what, they're not called samovars, that's for tea. Like one of those big coffee dispensers they have in motels. Sorry, Peyton. Ah, yeah, let's. So, top one is no tryptophan, and it's on. Trying to make sure you don't get the glare. So this is why it's repressible. It's always on unless it's told not to be. To turn it off, let's change the angle of the screen a little bit. To turn it off, tryptophan must be present because it changes the shape of the TRPR repressor. It binds to the TRPO operator and it prevents RNA polymerase from binding. So basically, stop expression dead. So again, no tryptophan, no TRPR binding, expression occurs, you know, as it did before. Presence of tryptophan, TRPR binds, blocks RNA polymerase binding and transcription, no expression as a result. So it's not trivial getting this stuff, right? It really isn't, because just trying to remember like what different things are with this kind of setup is really tricky, because there's a lot of conditionals, right? This must happen for this to happen, for this to happen, right? It's a lot better to try and think about it from terms of what needs to be done, right? In this case, if there's tryptophan present, it means that tryptophan is not being used faster than it's being produced. You don't need to turn chorismate into tryptophan, right? So you don't need this operon on. But typically, tryptophan is used as soon as it's produced, right? Because proteins are continually being made for whatever jobs those proteins do. And so tryptophan is continually being incorporated into proteins as soon as it is produced, right? So in that case, the TRP operon has to be on all the time, essentially, right? So it's really about kind of what is normal. In bacteria, it's normal that they're not on lactose, right? They're usually on one hopes, a food source that is more easily used than lactose. But when they are, and no glucose is around, they really need it. But when it comes to production of amino acids, at least ones that they can't get from their environment, one can assume that they always need them, right? It's not a context specific kind of deal. It's just a regular, well, we always need this stuff. And so for the trip operon, it's more a case of, well, if we don't need it, then we turn it off, right? Rather than we do need it and it's available, so we turn it on, right? So again, think about the kind of context, right? And so in that context, it makes perfect sense that when tryptophan is present, you turn off the trip operon, right? Because you just don't need it, right? So it's a really good way of make sure there's enough tryptophan around to be able to do what needs to be done, right, in terms of translation, but no more than that because it's an expensive thing to do, right? So if, I don't know if they can even take tryptophan up from the environment, but maybe they come across a nice juicy source of tryptophan. Perhaps they've, found someone's burger like underneath a barbecue joint in the state park, right? Then maybe they don't need to produce tryptophan, right? So, and they can get it from somewhere else. So they're going to basically shut down the trip operon. Or, nutrient levels are low, right? They have to go into some kind of dormancy, right? When they're not going to be producing lots of protein. So again, tryptophan levels rise because it's not getting used. And so you shut down all the stuff that you don't need anymore, right? So bacteria are astonishingly good at only doing what is absolutely necessary metabolically to be successful, right? They don't do any little bit more than that, right? Because if they do, then, bacterium next door, which does a better job, is going to outcompete them, right? Because there's typically a limited source of nutrients. And so the ones that are best able to use it are the ones that are able to divide and pass their genes on to the next generation. So bacteria are very, very good at adapting themselves to their environment and changing environmental conditions. And really, a lot of how they control their gene expression should be viewed within that context. Okay. All right. Now it's going to get a little crazy. This will, oh, no, not that one. Not that crazy yet. This one. Okay, so let's get the chat back. Oh, chat, please come back. I'm trying to make sure I can get the chat. All right, I can see that. Now I can't see. What have I done? I don't know. Bollocks. I'm sorry, still trying to figure out. Where's it gone? Here we go. I really don't know why Zoom does this. It's a real, oh, there we go. We've got it back again. Real pain in the backside. The chat disappears whenever I share a screen. So that's relatively straightforward, right? And this picture here does a good job of essentially what I drew on the whiteboard, right? We have this negative feedback loop of the product of the trip operon essentially causes the repression of the trip operon, right? So it's a feedback loop, a negative feedback loop. Now we go on to the fine tuning. This is called attenuation, right? And there is a, pardon me, attenuator region, and that's here, right? So if you look at this, let's kind of zoom this in a little bit. Right, this region here, called the 5' UTR, which is just upstream of the first structural gene, right? This is really, really, really important in attenuation, right? And the deal here is RNA shape, right? And so this is where it gets a little nuts, right? This is a kind of a blown up look at this, right? So let's make sure this is out of the way. And I take as much time on this as you need to, right? Here's the start of our first structural gene, TRPE, over here on the right. Over on the left, which isn't pictured, is the TRPO operator and the TRPP promoter, right? So if tryptophan is not present, or at least not present at higher levels enough to cause TRPR to bind, we're going to get transcription, right? So the RNA polymerase is going to bind to the promoter, it's going to move through the operator, and it's going to start transcribing this, polycistronic mRNA, right? Keep that in mind. Now, what is a key characteristic of gene expression in prokaryotes, particularly when it comes to transcription and translation? Here's a little, I think that was probably actually on the exam, if I remember rightly. What's one of the key characteristics? Loops, yeah, we've got loads of them here. What else? One of the keys is here. What happens in prokaryotes as soon as an mRNA starts being transcribed? Or very shortly afterwards, anyway. What happens next? Right, we get ribosome binding. And what do ribosomes do? They start translating the mRNA, right? So the fact that in prokaryotes, transcription and translation are coupled, right? They're linked together. Yeah, that's good. I got what you meant. Is really, really important. And that's a key part of attenuation of the trip operon. It's really, really critical. Okay, so keep that in mind, right? As soon as basically we've got RNA polymerase starts over on the left, kind of goes through the operator, starts transcribing this 5' UTR, right? It's going to go dang, 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 all the way through here at whatever speed that it normally goes through, right? As soon as that ribosome binding site is exposed, a ribosome will bind to it and will start translating this 5' UTR as the RNA polymerase transcribes it, right? That's how this works, right? It is really, really, really important. Okay. So that's one kind of key characteristic, right? And let's write these down because it never hurts to have this stuff kind of codified, right? So that we got some kind of record for it. Oh yeah, you're being very cute. Sorry, I'm busy. My cat's going to be very sad when this course ends because she expects me to sit here petting her basically for several hours a day. Okay, I'm going to try and manage glare as best as I can. So we have, and I'll come back to the PowerPoint because the visualizations are a lot better essentially than what I'm able to produce. So we have our TRPP, we have our TRPO, and then we have our, let's put oops, if I reach over here. Right, there's our TRPE, which is our first structural gene. And this is all our leader sequence, right? So this does not code for a protein that does anything in the metabolism of tryptophan, right? Its sole purpose is to allow the control of expression of the trip operon, right? Or rather, the attenuation of expression, right? And so one of the key is we have two trip codons, right? There. And we have a bunch of loops. Or rather, loops that can form, right? Which ones form depends on whether or not we have tryptophan present. Does that make sense? Right, this is going to be a little tricky to kind of get because it's a tricky concept to absorb. Right. So the first thing is that essentially we're going to have, let's see if we can keep colors the same. Here's our RNA polymerase, right? Again, it hasn't started transcribing the TRPE gene yet, right? This is really important. If I can spell it, here we go, right? But it has, let's use blue. It's already transcribed some of the leader RNA, or mRNA really, if I'm going to be particular, right? Because nothing's stopping it over here from doing so. Yep, does that make sense? Okay. Give me a shout if it doesn't. And so, yeah. Now we're going to have our ribosome bind to it. Okay, because transcription, translation, and prokaryotes are coupled together. Right. Any questions so far? Okay. I'm going to see if I can do this all on this teeny tiny little whiteboard, but we'll see. Okay, so bear with me. I'm not going to draw out trip P and trip O anymore because it's not relevant anymore because transcription, sorry, has begun. Now in here, remember I'm just going to label them with T's. Those are our trip operons. Paget, codons. I always get confused between the two, right? And so the ribosome, as it comes through and translates, is going to need tryptophan to continue translation. This is how it works. If it doesn't have tryptophan, it's going to pause here. Does that make sense? Say yes or no. No is good because it means I'll be able to explain it more. Yes is also good because it means I've explained it well. Oh no, both are good. Okay, so this is the key, just here, right? So because the ribosome and the RNA polymerase are they're not linked physically, but the whole process is coupled together. And because we have two trip operons here, tryptophan, right, the product of the trip operon expression, tryptophan is going to be needed for this ribosome to continue producing this leader protein sequence, right? If tryptophan is not present, right, because it's being used faster than it can be being produced, this ribosome will stall, it will stop, right, because there's no trip tRNA to be able to incorporate tryptophan into this protein. So whether or not tryptophan, the amino acid, is present, that controls whether or not this ribosome can translate this leader mRNA or not. That's the key to how this works. Did that help, Peyton? Cool. Okay, so let's continue down here, right? So first I'm going to do the no tryptophan, right? So remember, no tryptophan at all. Trip R cannot bind. Transcription begins. This whole process starts, right? So okay, let's draw this out again. And why do I use so many colors? Okay, so RNA poly is going to be a little bit further down. Not the end yet, but further down. And basically, crab sticks. We're going to, how are we going to do this? Here's our ribosome. Our ribosome is going to stall. It's going to stop. I haven't drawn out the rest of the mRNA yet because I want to show what the consequences are, right? So essentially, no tryptophan equals no trip tRNA, right? Because there's no tryptophan to stick onto a tRNA by, the amino acetyltransferase for that tRNA to come into the ribosome and donate its tryptophan to the growing polypeptide, right? So no tryptophan, no trip tRNA, ribosome stalls. And what we mean by stalls is it stops, it pauses, right? It cannot continue any further. If tryptophan does become present, then, it can, but at present, it's not. Right? And so now we get into the loops. So now we get loop 2 and loop 3, or region 2 and region 3 is probably a better term, right? It looks like that. Because this region is region 1, right? Where the where we at? The trip codons are, that's called region 1. Yeah, I'm pointing to the right thing. Okay, so when tryptophan is not present, the ribosome stalls on region 1, and as a result, the mRNA of region 2 and 3 forms a loop, right? This is called, This is called the Sarah Connor loop. Let's see if you get the reference. Does anybody? Someone did at least, right? And the reason is it's called the anti-terminator loop. Ta-da! Sorry, my joke's terrible, as my kids tell me at least. Oh, yeah, probably shouldn't repeat that one. So what happens now is nothing, right? RNA polymerase continues into the structural genes and it makes a polycistronic mRNA and oh yeah, sorry. Yeah, it's just one of my bad jokes, Angela. Don't worry, you didn't miss anything. Yeah, it's the Sarah Connor loop. It's the anti-terminator loop. Actually, I want to watch the most recent one. It's supposed to be pretty good. I like the Terminator films. Right. So again, it doesn't. Hey, stop that. All right, my cat's coughing up a fur ball. You done? Yeah? Yeah, I think she's good. Anyway. Never seen him? What? Classic American cinema history there. You won't get all kinds of film references from, 30 and 40-year-olds like me. So, essentially, no tryptophan. Ribosome stalls on these trip codons because it can't, fill them, right? Can't progress beyond them. That then allows RNA polymerase to go on a little bit further and forms this 2-3 loop. Okay, does that make sense? And because of that, essentially the RNA polymerase is unmolested and continues on its merry way and transcribes the whole operon. That's what you have with no tryptophan present. Does anybody got any questions about that? Oh, okay. Let me take a picture of that. And you want to keep going or you want to take a short break? Short break? Any other requests? All right. So, because the next bit is actually kind of tricky, it'll be good to do it with a fresh brain. So let's come back in 15 minutes, continue at 12:30, and we'll go over what happens when tryptophan is present, right? You can actually look ahead if you want in the PowerPoint, and you'll be able to tell me. Okay, so I'll see you. Hopefully that will be after the break. So talking about pardon me, the rest of attenuation will probably be about another 10 minutes or so, and then we'll start PowerPoint 18 on eukaryotic gene regulation. Okay, so I'm just going to pause the recording. I'm going to go stretch my legs. Let's get this off. Actually, all right, what I'm going to do is leave most of this up. Instead, what I'm going to do is I'm going to take off the bottom part, right? So let's just get that cleaned off. I'll give you some love later, Cat. Okay, so same deal through here. Trip E. Okay, so what happens now, let's get this down here a little bit. Maybe actually, it's kind of thinking on my feet. Let's use both of these. Okay, so again, some noise back from the mic. Okay, so we have our RNA pol, and we have our ribosome. So if let's put this up. Okay, so the situation now is not that we have a lot of TRP or tryptophan present, because if there's enough tryptophan present to bind to the TRPR repressor, then you'll shut down the TRP operon expression completely, right? It is pretty much absolute. So this is a situation if we have some tryptophan present, right? And this is not a defined amount. It's somewhere between nothing and a lot, right? And so if you have some tryptophan present, remember these two TRP codons. Now what will happen is we have our mRNA being produced, right? And here's our region 1, right? Remember, we've got 4 different regions. In the last one, and this is region 1 is where the TRP codons are. In the absence of tryptophan, the ribosome will stall on those TRP codons, right? And that will result in the formation of the 2,3 loop, which doesn't do anything to RNA polymerase at all, right? Keep that in mind. So now, however, there is some tryptophan present, and there's enough for the ribosome to be able to pass through. And essentially, it's going to go on a little bit further. Let's get the ribosome up. And it's now going to move at the same with the same pace. It's going to cover up region 2 by the time region 3 and 4 are produced. And then there's 4 being produced over here, right? So those are the 4 different regions. With tryptophan present, we get our translation, right? You can kind of imagine that being sticking out the top. Right? I'm going to make sure you can see this well. And most importantly, the ribosome now covers region 2. So the anti-terminator loop, the 2,3 loop, cannot form because the RNA needed to form a loop with region 3 isn't present. It's not available to base pair, right? So the 2,3 loop cannot form. And so the consequence of that, and there's a kind of a UU, and if you remember this from intrinsic termination, right, of gene expression in bacteria, this poly U, whatever it is, however many there are, region will slow down the RNA polymerase, right? It'll cause it to pause a little bit. Everybody with me so far? So just to recap, tryptophan present, ribosome continues translation. And again, that protein doesn't matter, it's irrelevant. And now the ribosome will be occluding, it will be over region 2. Got it? Now we need to see what happens with these two. This is where all of the action happens essentially. And so now we're going to have, we have our ribosome, we have our region 1 and 2, and now we have our terminator loop. So this is 3, 4. This is our terminator loop. Right. And what it does is it basically gets into where the RNA polymerase is, like that RNA DNA hybrid, and it forces the RNA polymerase off of the DNA. Sorry. And so, as a result, that RNA polymerase does not continue into the TRPE structural gene, and transcription is terminated. So just to kind of emphasize the point, and there's the UUUs are down here. Oops, lost something. Right. And so this forces RNA pol off the TRP operon DNA. Right. So now you can see that, no, sorry, I'm being a little bit slack. This is the ribosome. The red is the RNA polymerase. And so that's why transcription and translation being linked in prokaryotes is not just an observation, it's a really important aspect of how they control gene expression. Right, because essentially what RNA polymerase, what the ribosome is doing here is that the ribosome is a sensor for how much tryptophan, is present. Right, just as the catabolite activator protein is a sensor for cyclic AMP, which in turn is essentially the indicator of glucose levels. Here, the ribosome is the sensor for tryptophan levels, because if there is no tryptophan, it pauses on the first region of the mRNA transcript. Remember, that's being produced at the same time by the RNA polymerase. And when it pauses on the first region, the 2,3 loop forms and transcription continues as normal. However, when there's enough tryptophan present for the ribosome to continue, right, from region 1 onto region 2, obviously not enough tryptophan to stop transcription entirely. Now the 2,3 region cannot form a loop. Instead, what will happen is the 3,4 region forms a loop, and in doing so, that's why it's called the terminator loop, it levers RNA polymerase off of the DNA at this triple or poly U region that it's transcribing. Right. So essentially, this deal here is a modification of intrinsic termination in prokaryotes. Right? Remember, in intrinsic termination, this terminator loop and the poly-U region are part of the gene, right? Usually in the 3' UTR, and that's what allows the bacteria to terminate transcription at the end of a gene, right? Normally, obviously, there's row-dependent termination as well, but this intrinsic termination is not at the end of the gene, right? There's probably something else at the end of the trip operon which does the same thing. This is essentially being used as a mechanism for controlling whether or not RNA polymerase continues on to transcribe the structural genes of the trip operon. And it will only do that when there's insufficient tryptophan to be able to translate this leader polypeptide, right? Because it'll cause the ribosome to stall. So the ribosome is really, in a sense, sensing tryptophan levels, right? Because if there's no tryptophan, there's no tryptophan tRNA, right? tRNA charged with tryptophan. And so this polypeptide cannot be translated. And as a result, this terminator loop will not form and transcription continues, right? And you can imagine, like, transcription continues, those structural genes are produced, tryptophan is produced, right? So now tryptophan is going to be incorporated into a tRNA, right? Charged onto a tRNA. Now this ribosome can continue translation, right? Because it has charged tRNA to make into the polypeptide, right? Over these codons. And now that anti-terminator loop cannot form, and the terminator loop forms down here, and that stops transcription. Now, obviously, you know, we can try again, right? Because there's not enough tryptophan to prevent transcription at all, right? So you can imagine, like, you know, a split second later or a few seconds later, the tryptophan levels drop again, right? Because the tryptophan that was present is now incorporated into this protein that's being made up here, right? So now when transcription occurs again, it comes through, now there's not going to be any charged tRNA, tryptophan tRNA. And so the ribosome is going to pause here. Terminator loop does not form. Transcription, RNA polymerase stays attached to the DNA. Transcription of the structural genes occurs. Enzymes produced. Tryptophan levels go up a little bit. Tryptophan gets incorporated into protein. Ribosome doesn't pause. Terminator loop does form. Transcription is stopped, right? So it's that constant fine-tuning, essentially, that occurs as part of this attenuation, right? So it's not a big-scale deal. It's a very much a tweak things as we go along, right? Just to maintain tryptophan levels at the, you know, or to provide sufficient tryptophan for whatever's needed. Questions? Cool. Okay, no worries. Does anybody have any questions about any of this? This is not trivial to get, right? Because there's a lot of, it's two different series of events, right? Which are conditional on different contexts, right? So have a think through this. We can go over it again beginning of next lecture on Thursday, just to recap, right? As we did with the lac operon. So again, just keep in mind what's going on. What do we need to do, right? What are the needs of the organism with regards to making tryptophan, right? And how do you best accommodate those needs, right? Because you can see that just switching on and off the whole operon using TRIP-R is going to be a fairly blunt instrument, right? So essentially, you know, you produce some tryptophan, but it's, you know, it's only enough for, I don't know, let's say one protein to be produced, right? But you shut down the whole thing. And then that protein is produced, and now you don't have any tryptophan in the next protein, it can't be made, right? Say whatever that protein was from, like ATP synthase or some kind of structural protein or whatever, right? So now you have to start the whole thing up again, right? And start producing the components and so on and so forth, right? So if levels are kind of fluctuating a little bit, you don't want to turn the whole thing off. You just want to dial it down a little bit temporarily, right, before it can turn back up again. That's the whole deal behind attenuation. I'm going to take a picture of that before I forget. All right, anybody got any questions? No. And again, look through, I mean, the images here are you know, quite neat essentially. And this has, come on, a really quite nice way of describing it in pictorial form, a lot neater than I can do it, right? So essentially this is what occurs, just what I've described, right? The ribosome is able to proceed through this region 1 into region 2, so this terminator loop forms, and that kicks off the RNA polymerase. Okay, so if no one has any questions, we can get going on PowerPoint 18. See, nearly done. It's only a couple more after this. Okay, so I'll probably be bouncing around a bit in here because the order's a little bit different from kind of how I would think about it. But essentially, in eukaryotes, there are many, many, many more steps at which expression can be controlled, right? So if we talk about gene expression as being the whole process that ends up with a functional protein, because that's really what we want. In eukaryotes, there are many, many different steps along that process where things can be controlled, right? They can be boosted, they can be reduced, they can be stopped altogether, right? And there are lots of different aspects, right? So one, we can change or control how much that gene is turned on, right? Or where or in response to what, right? So we're talking about transcription. We can control mRNA processing, right? How is that transcript spliced? We can have alternative splice forms, right? Which we'll talk about in a little bit. How long does that mRNA hang around for, right? So if that mRNA is around forever, then you'll get a lot of protein out of it, right? Because the ribosomes will be constantly translating it. If you don't want very much, or you want it only for a short burst, then you get rid of that mRNA very quickly, right? 2, 4, maybe. Control of translation, right? So you can control how efficiently translation occurs. 5, we can control how that protein is processed, right? There are an awful lot of proteins, because eukaryotes have organelles, right? Prokaryotes don't. There are an awful lot of proteins which are not in their final form as translated. Sorry, didn't want you to get that through your microphones. Part of the reason being they might not be in their final form, right? They might need extra processing to attain the final shape, or it may be that they're not needed at that point, right? So they're going to be stockpiled, and so you don't want that protein to be active because it's not yet needed, but you will need it at some point, right? Insulin is actually an excellent example of that. So your beta cells in your pancreas produce insulin all the time, right? But they don't release it all the time. They only release it in response to blood sugar levels going above a certain threshold, right? Part of that kind of cellular thermostat, right? Or bloodostat or glucostat, I guess would be the word for this. But when it does need to release it to tell muscles to take up glucose from the bloodstream, it needs to produce a lot of it, right? And this whole gene expression process takes a long time. And so for insulin, it's produced in one form, it's processed, kind of chopped and diced, and then it's stored until when it's needed, right? Stored in those beta cells. And so the process of gene expression in the sense of producing a functional protein is not yet finished for insulin. It'll only be finished when that storage vesicle fuses with the kind of the beta cell membrane, and then you release that insulin into the bloodstream. Then it can actually do what that protein was intended to do. Right. You can also get rid of proteins, right? Maybe you don't need them anymore. Maybe they're damaged. Maybe you just produce more than you need. So you break them back down again, right? There's all kinds of different things going on. And this is actually like a really nice graphic, I guess, right, which talks about that control. Also, I forgot, obviously, that DNA has to be available for transcription, right? So we're going to be modifying the chromatin either by histone modifications or histone acetylation or DNA demethylation, right? Because that also blocks transcription. Before we can even think about turning a gene on, right? The DNA that encodes that gene has to be available. And then these are all the kind of the final steps, right? So each one of these steps is basically available, right, to be modified or controlled in some way to affect the overall outcome or output of that gene, right? So again, it's not just control of transcription, right? Though that's a very, very big part of it. It's all of these. It's the sum of these different processes, right? So I want to talk about, let's talk about chromatin first. I really do need to reorganize these at some point. Okay, and so there's lots of different ways to think about chromatin. And this is a big old chunky PowerPoint, and well, it goes all the way down here. Lots and lots of pictures, right? So we're not going to talk about all of this today at all, right? This will we'll continue this on Thursday, right? So we don't overwhelm me too much. Where was I? Here we go. So essentially, in terms of chromatin, we have two different levels, right, at which we're going to control this. One is the very big scale level, right? If you remember, I I think it was the last section that we talked about, pardon me, before the exam. We talked about like the ends and the centromere being heavily condensed, right? All heterochromatin. So on the very large scale process, you can open and close big regions of DNA. We're not talking about region for one gene. This could be big enough for hundreds or maybe even 1,000 genes, right? Typically not that big, maybe. But anyway, certainly you can control the availability of very large numbers of genes at once. And so typically, not always, there are obviously always exceptions to this, but typically or quite frequently, if you have a particular process that you want to control, quite often many of the genes involved in that process, let's say blood vessel formation, right, which generally speaking you only do during development. I'm not talking about increasing, vascularization like you do with exercise or in tumors, talking about actually forming the things in the first place, right? So many of the genes involved in initial blood vessel formation will be grouped together in the same part of a chromosome, right? We're not talking about right next to each other. We're talking about within the same general region. So when it's time to begin vascularization, which is, oh shoot, when is that in development? That's probably, I think that's around 12 to 14 weeks. The heart forms first and then blood vessels kind of form after that. I'd have to double check when that happens in humans, but kind of about beginning of the second trimester, I think, by and large. I think you can get a heartbeat still in the first trimester, but it's much more reliable in the second one. Anyway, so up until that point, until heart formation begins, you don't need to express those genes, right? So that whole region of the chromosome will potentially be condensed, right? So it's shut off. The same thing after vascularization has ended, right? And you've got all of the basic, veins and arteries that you need. You don't need no more, right? You'll just build off of the existing ones you have, which is a process called angiogenesis, right? So all of those genes, their information is no longer needed, right? One hopes. And so now you can basically condense that whole region down and turn off all of them at the same time, right? You don't need to turn off each individual one separately. You're just shutting the door on all of them, right? Obviously, that's a lot more complicated, right, than that. But that's a good general overview way of thinking about it. You can also control access to individual genes, right, which is what this graphic is a good way of describing, right? So on individual genes, you're still going to have protein complex with DNA, right? That's the whole point of the protease K digestion in DNA extraction, is to get these proteins away from DNA so they don't co-precipitate. And so you can now control how tightly around those histones that DNA is bound, and you can even slide the histones up and down, right? The DNA to block or open up access to particular elements of that DNA, right? So this is a really good example because we've got the Tata-Box binding site, right, which is Tata-Box binding protein. Yeah, something like that. Tata-Box protein binding site. Yeah, whatever it is. This is where the Tata-Box binding protein binds, right, to help assembly of RNA polymerase. If it is tightly wrapped around a histone complex, it's not going to be available, right? That protein is not going to be able to recognize it and bind to it to allow RNA polymerase to assemble. Same here, we've got transcriptional activator protein binding site. If that is occluded, even if this TBP was open, it still wouldn't matter because we wouldn't be able to activate transcription, right? So this conformation results in no transcription or very, very low transcription. Here's another good one. Is this what I want to? Oh yeah, here we go. Right. And so we can control transcription even in the absence or without consideration of any other proteins that are involved in controlling transcription, like the activator proteins, mediators, transcription factors, any of that. Even in the absence of those, we can restrict expression, right? We can repress transcription. Oh, I probably better talk about epigenetics, right? And so this essentially the modification of histones and DNA in a way which doesn't change DNA sequence is called epigenetics, right? So you're able to do so, and this is a stable heritable change, right? So you do inherit epigenetic modifications from your parents, right? Especially from your mother. And it does not change the DNA sequence, right? This is actually a pretty trippy thing to think about because it means that you're inheriting information from your parents, which is not information encoded permanently into your genome, right? So it's much more of a kind of conditional information, right? Only lasts for a couple of generations. And so we can make these modifications at a bunch of different levels, right? We can modify histones to make them bind DNA more or less tightly, right? So we kind of make that heterochromatin versus euchromatin, right? We can modify DNA itself, which is by methylating cysteines or cytosines, sorry, not cysteines, those are the amino acids, modifying cytosines, right, called CPG islands. And we can also remodel chromatin, right? We can shift these histones around, right, to create open areas or compact areas which are not available for transcription. And so these chromatin remodeling complexes, it takes a lot of energy to do this, right? We use ATP for. Essentially, we can do a bunch of different things, right? We can remove histones entirely, right? Just kind of kick them off, right? Which will obviously allow that DNA to be accessed very readily, right? We can also slide these histones kind of up and down like an abacus, essentially, along the DNA. So we kind of shuffle them over here, that then leaves this region open for transcription, or we can pile them back in and close things off completely. And we can also rotate the how the DNA is wrapped around the histone. So if you imagine we have histones like so, right, we have DNA wrapped around each one. If you rotate them slightly, then some of the DNA that was originally bound to this histone is now going to be available in between the two, right? And so and then you kind of basically go back again. Anybody got any questions? And again, this is really like the central point to get across is that all of your cells have the same information in them. All of them, right? They all have exactly the same DNA, apart from your red blood cells, which don't have any. But they are very, very, very different. Good videos, not off the top of my head, but I'll have a look. Let's write that down on my. When in doubt, I typically look at the WEHI, WEHI videos, those ones of the molecular motions, those are generally very good, but they don't do everything. So I'll just make myself a note. Feels like I'm carving this into my hand. Don't like these gel pens very much. They're a little bit painful. So yeah, this one's pretty obvious, right? So we have our chromatin, our DNA protein complex here. Our remodeling complex comes in, uses some energy, and simply kicks one of those histone octamers out, or maybe even two of them. Now that basically leaves that DNA flapping in the breeze, and transcription factors, RNA polymerase, so on and so forth, are now able to physically access that DNA. Right, a lot of this is actually it's biomechanics, right? It's not so much biochemistry, but more what things can physically get in there, right? And I mean, it's like kind of jamming into a crowd to get to the bar, right? If there's more space, then you can get to it more easily. Not that we're able to drink in bars anymore. And it's a lot of what we're talking about here is really like the mechanical aspects of biology, right? Physically moving things around and allowing, creating space or not, as the case may be. This one is a really nice, so I'm going to zoom this up a little bit so you can see it more readily. So if you remember, where are we at? Come on, one more. Here we go. This one, these key DNA binding sites are obscured, essentially, right? They're inaccessible, right? So we can use these chromatin remodeling complexes to basically slide or rotate these histone octamers to create some space, to be able to open up that DNA, right? So if you look at this one up here, right, we have our activated protein binding site here, right, if you remember from before, and we have our Tatabox binding protein was originally here. So now we bring in our chromatin remodeling complex and we rotate the DNA around. So really what we're doing is it's like we're kind of pushing beads on a string, right? And so we can slide them in general, or we can like rotate them. It's the same outcome. And in doing so, DNA is obviously, it's still bound to those histones, right? We haven't removed those histones. We haven't kind of ballooned out the DNA so it's more accessible. What we've done is we've just rotated the DNA around, and now we have these two binding sites are now available, right? Before they weren't, no gene expression. Now they are. Now we're able to bring in our Tatabox binding protein, right? We've got our transcriptional activated protein. We've got our TF2D, our transcription factor that brings in RNA polymerase. Now we can bring in RNA polymerase. And what RNA polymerase does, it's a bit like a snowplow, right? As soon as it's able to bind, it will move along and it will just basically push those histones off of the DNA, right? And they'll come back again afterwards as it goes through. But RNA polymerase will, as long as it can bind to the DNA, right, as long as that DNA is accessible and available, it will force its way along the DNA strand and it will transcribe it as it goes, forcing those histones off. Okay, anybody got questions? Okay, another way, is this one that does that? Yeah, this is a good one. another way in which you can, hey, quit it, modify or control access to DNA is by modifying the histones or the DNA, right? So there are two deals going on here. We've got acetylation equals basically pro gene expression. And that happens to histones to make them less charged. Right, so essentially you see these tails on these histones proteins. These histone tails are charged. Right, typically they're due to lysine residues is the main one. So they're positively charged. These histone tails attract DNA more tightly because of the ionic bonds between the positive tails and the negative backbone of DNA. And that DNA is bound quite tightly to those histones, right? So it's not available for proteins to bind to it, right? It's very, very tightly bound up. Now, if you acetylate those lysine residues, essentially what you're going to be doing is putting a small functional group onto that amino acid, and that stops it from being charged. Now the DNA is able to relax, essentially, from those histones. The histones become further apart, and then instead of being like tightly bound to the histone octamer, the DNA kind of relaxes off of that histone octamer. Both of those make that DNA more available for gene expression, right? So just modifying these histones, right? So you have histone acetylases, right? Because they add acetyl groups or acetyl groups to those proteins, and then that makes the DNA more accessible. You also have the opposite, right? So you have histone deacetylases or HDACs, and those are anti-gene expression. So they remove those acetyl groups or acetyl groups, and that causes the DNA to compact back down onto the histones and become less accessible. So we have this constant like seesaw balance, right, between open chromatin or particularly acetylated histones and deacetylated and methylated or unacetylated histones and closed chromatin, right? And so for any given region of chromosome, there's this dynamic switching between those two, right? We're not talking about the big scale changes of chromatin, but this can actually also happen at like the smaller scale too. And there's also methylation. And that typically is anti-gene expression. And, capital letters, can happen to both histones and DNA. As you can see here, these tails are methylated. I'm not entirely sure what that does to histones. I think it causes like a conformational change. It may make the lysines less able to be acetylated as one possible explanation. I'm not actually 100% sure. Again, I know an awful lot about molecular biology and the like, but there's still an awful lot that I don't know. And some of the peculiar, wrinkles of this particular process I just don't know them. I do know that there are different lysines in each tail, and depending on which one gets acetylated or not has an effect on the overall outcome, but the details are pretty obscure, to be honest. Okay, so that's for histones. You can also inactivate DNA. And so the deal here is if you methylate DNA, and this is always the Cs, the cytosines, you change its appearance to proteins. And so you have your double helix, and transcription factors or transcriptional activator proteins recognize a sequence of that DNA, and they will bind to, and they will recognize and bind to a very specific sequence and shape of DNA. So if you modify that DNA, right, and this isn't a permanent modification, this is potentially transitory, right? If you modify that DNA, it doesn't look the same to those proteins. And that's essentially what DNA methylation does, right? It adds these methyl groups or methyl groups. There we go. Oh, no, that's a different question. Where are we at? Blah, blah, blah. Here we go. Right, and that's basically adding this methyl group to cytosine here. Is there a better graphic down here? Yeah, more or less. Essentially, these changes the shape, and also these are called CpG repeats because it's a cytosine phosphate group and then a G. And you tend to find more of these in promoter regions, right? So if you remember all the way back now to DNA replication, right? This DNA methylation was how the mismatch repair determined which is the new strand and which is the old strand, right? So we talked about this already. But once DNA replication is finished and methylated, both strands essentially look the same. So these CpG repeats tend to be more common in promoter regions. And so what that means is that if these regions are, that'll do. Now, I need to go back through and fiddle around with these PowerPoints a little bit. Essentially, if those promoters are heavily methylated, then the transcription factors that are required to turn them on won't recognize that DNA. And as a result, DNA expression will be suppressed. Now, the interesting thing is, and this is kind of taking it back to the big picture again, you tend to find, so there are, as we've talked about before, lots of ways in which you can promote or inhibit tumorigenesis, right? The formation of cancers. And there are a whole family of different genes which are called tumor suppressor genes for obvious reasons, which prevent tumors from forming. A lot of that's involved in controlling apoptosis or cell death. And if you look at patients that have cancer, quite often what you will find is the promoters of those tumor suppressor genes are heavily methylated. Now, we don't actually know if that methylation leads to cancer or if it's a consequence of those tumor cells preventing their own demise. I don't actually really know. But it's a very strong correlation, right? Essentially, the very genes which are needed to control tumors or to reduce their growth. Hey, quit it. Sorry, cat issues. Those tend to be suppressed in tumors. And they're typically suppressed by DNA methylation, right? So you can actually look at the DNA from cancer sufferers, and you'll see a particular kind of pattern, I guess, of methylation. So again, kind of trying to take it back to the bigger picture. This obviously isn't just a cancer deal. Obviously, it applies to all kinds of different genes, but it's just another way of thinking about it, essentially. There's also a feedback between DNA methylation and histone methylation, right? So quite often these methyl groups or these methylated cytosines, they also serve to control histone methylation and acetylation too, right? So quite often where you have these methyl groups, a histone deacetylase will be recruited, an HDAC that we talked about just a moment ago, which will result in the deacetylation of that histone and potentially an addition of a methyl group, right? So not only do you inhibit gene expression from that particular promoter, but also you can then have a positive feedback loop where you further shut down access to that DNA by modifying the histones too, right? And so, and here's just another illustration of that, right? So here's our histone, right? This is histone 3 lysine 9, right? And methylation or DNA methylation results in the interaction between DNA methyltransferase and this histone methyltransferase, which further shuts down gene expression, right? So it's kind of like a cascade effect, essentially. Right, what else do we have to talk about in terms of methylation? I think that's pretty much it for talking about chromatin. So remember, in terms of chromatin, we have two different scales, or kind of a range of scales from one extreme to another, right? We have the whole chromosome scale, where we can shut down entire regions of that chromosome, but also we have the very focused kind of scale where we're controlling access of proteins to the regulatory elements of that gene, and on a gene by gene basis, right? And so remember again that acetylation is typically, and it only occurs to histones, is typically pro gene expression, right? Because you get this more relaxed DNA binding right here, more accessible DNA, and also the kind of condensed heterochromatin tends to relax, right? Not 100% sure how that works, but I do know that it does, right? So that's generally, we want to open up the chromatin, and we also want to open up the DNA to be accessed by whatever regulatory proteins need to access it. Methylation is the opposite, right? So histone methylation essentially reverses this process. It causes DNA to be much more tightly bound, causes those nucleosomes to clump together more tightly, so you start shutting down whole regions of a chromosome, not just one gene. And also DNA methylation, even if that DNA is accessible, it will be more poorly recognized by transcription factors, and also result in the methylation of histones, right? So you have this kind of dynamic balance between kind of like the closed heterochromatin where nothing's available to the more open euchromatin. And you can do that on all kinds of different scales, from like, tens or hundreds of genes down to individual genes themselves, right? So that's a really, really big part of how gene expression is controlled in eukaryotes. That's before we get to anything else about turning genes on, about how we're splicing them, how we're processing the mRNA, how stable the mRNA is, what we're doing to the protein afterwards, right? Before we can get to any of that, we need to control one way or another the chromatin status. And the crazy thing is that a lot of these differences are heritable, right? They've actually found that people, particularly women who smoke, particularly smoke heavily, that results in especially DNA methylation changes, and that is then inherited by their offspring and potentially their offspring's offspring, right? So even if you don't smoke, but your grandmother smoked like a chimney, right? You're still potentially at risk of the the side effects of smoking. Isn't that nuts, right? And it typically peters out after, like the third generation. But certainly, whatever your particularly grandmother, um, whatever your grandmother on your mother's side experienced, you will bear some of the consequences for that, one way or another, right? So we're not just the sum of our DNA, not even just our nuclear DNA and our mitochondrial DNA. We'll also inherit some of those changes to that DNA which occurred due to the experience of most likely your maternal grandmother. Typically on your mother's side, typically, doesn't have to be though, but most of these transgenerational studies tend to find effects through the maternal side than the and then the father's side. Hey, this isn't prescriptive, right? It's not like uh, you know, grandmother Josephine is like sitting in this cloud of tobacco smoke 24/7 and you're screwed, right? It's not prescriptive, but it's part of the information that makes you who you are, like it or not, right? Yeah, Wally, hell, that's impressive. Not in a good way, but yes, I mean, that's part of how these traits are inherited. It's not just the DNA that you inherit from your parents who inherited from their parents. It's also whatever modifications will manage to make it through uh, all those rounds of DNA replication to end up, as you essentially. So it's pretty crazy. These epigenetic modifications are also the reason why old identical twins no longer look identical, right? So, when they pop out, identical twins are, indistinguishable from each other. And typically until that's the case until, they get past their teens. Obviously, they dress differently, had different haircuts, stuff like that. That's a given. But physiologically and in terms of appearance, identical twins will look identical at least into their late teens. But as soon as they start experiencing different things, say one of them smokes or one of them doesn't, one of them has a really shitty time where they're super stressed out all the time and the other one doesn't, whatever. There are lots of different ways in which our environment affects our DNA, or chromatin. Over the course of your lifetime, those changes accumulate. And in identical twins, it leads to slight but significant change in those two individuals. So they are no longer 100% identical, right? Because they have had different experiences, and their DNA, which is essentially the sum of their DNA sequence, which is identical, but also the DNA modifications and the histone modifications is no longer the same. And so if you look at identical twins in their 50s, they do not look identical anymore. They look very, 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 very similar, but you can actually tell them apart if you weren't told who is who. Isn't that crazy? biology is pretty awesome as far as I'm concerned. I mean, that's why I'm a biologist, right? Cool. Okay, I think that's more than enough for today. Let's take a break until, let's make it 2:05, just so we have enough time to throw some food down our necks. And we will meet again for lab at 2:05. Oops, let's type that up. If I can spell meat properly, because I am absolutely famished. All right, and then next class on Thursday, we will talk about all this other jazz, like splicing. We'll talk about, stop it, RNA interference or control of mRNA stability. We'll talk about some other funky stuff. We'll talk about cis-regulatory elements, which are all the way down here. Oh no, not that far, that far. Right. And essentially like all the other levers and dials that we can control in terms of gene expression. Right. So we'll finish this up on Thursday. All right. Thanks very much, everybody. I'll see you in half an hour.